Hello, my name is Luminica Moroz and I am part of Lucas Schell's lab at Science for Life Laboratory in Stockholm, Sweden. What I'm going to talk to you about in this tutorial is how you can use our new software tool called GradientOptimizer to calculate in silico optimized gradients. In silico optimized gradients are a type of nonlinear gradients which we proposed in an article that was recently published in Analytical Chemistry. I strongly recommend you to read this article before trying the Gradient Optimizer tool. It will give you insight in the different types of nonlinear gradients that you can calculate and also the characteristics and advantages of each such gradient. So please read this article before watching this video. Also, in this tutorial I will use Elude, which is a machine learning based prediction method developed in our group. I would also recommend you to take a look at the article that describes Elude, which was published in the Journal of Proteome Research. In this tutorial, I will start by describing the main ideas underlying the use of nonlinear gradients, and then I will move on in explaining step by step how you can calculate an in silico optimized gradient using the Gradient Optimizer software tool. So first of all, why do we need nonlinear gradients? As you know, in chromatography, when we separate peptides, we use most of the time a linear gradient. That means that if we would plot the percentage of organic solvent as a function of time, we will obtain a line, like you see in this image. The problem with using this kind of linear gradients is that they produce an unequal distribution of the peptides over the run. Here in this figure, you can see a distribution of the peptides that we obtain in one of our runs. On the horizontal axis, we have observed retention time. On the vertical, we have number of peptides. As you can see here, there are much fewer peptides eluting in the beginning of the run and many more in the middle of the run. Following this observation, what we wonder is, could we come up with a different type of gradient function, possibly nonlinear, that would give us an uniform spread of the peptides over the run? And this is the main idea underlying nonlinear gradients. We want to obtain an equal, uniform distribution of the peptides across the run. If you would look at the article where we propose nonlinear gradients, we described two different types of such gradients. Here I will introduce one of them, which is called in silico optimized gradient. The in silico optimized gradient aims at giving an equal distribution for the theoretical peptides from an in silico digest of the proteome of interest. In this figure, I'm showing the main steps of calculating such a gradient. We start with the set of peptides that were produced using a linear gradient, so peptide identifications that were resulted with a linear gradient. We use these peptides to train a retention time prediction model, which can then be employed to predict the retention time for all the theoretical peptides from an in silico digest of the proteome. Since the predictor was trained with data that was found using a linear gradient, this distribution will be fairly skewed. Now, however, from this distribution we can calculate what would be a uniform spread of the peptides. So how would we flatten this distribution so that it becomes uniform? Once we did this, we can actually calculate the gradient function, the nonlinear gradient function that will provide us with this distribution. So we started with a set of identified peptides with a linear gradient and a set of theoretical peptides, and we end up with an optimized gradient function that will provide an equal distribution of the peptide across the run. This is the main idea behind calculating in silico optimized gradients. Now I'm going to move on to explaining step by step how you can calculate such a gradient using our software tool. The first step is to make sure that Java is installed on your computer. This is a MacBook, and an easy way to do that is to open a terminal window here and type Java -version. If Java is installed on your computer, then you will see a message similar to the one displayed here, Java version 1.7. That means Java 1.7 is installed on this computer. If, however, you don't have Java installed, then an error message will be displayed. In such a case, it is easy to download Java and install it. It is freely available on the internet. If you use another operating system, like Linux, you can check your Java version in a similar way as shown here. Just open a terminal window and type the command java-version. If you use Windows, you can just Google checking Java installation in Windows. It is also straightforward to check your Java installation. Now, once you made sure that you have Java installed on your computer, the next step is to download and install the retention time prediction method elude. And I'm showing here two different links from where you can install elude. I'm not going to go into many details how you do that. I'll just show you the two links that I'm displaying there. The first link gives a list of distribution packages, both for Percolator in the upper page and for elude in the lower page. You just need to choose the package corresponding to your system and install it. It is straightforward to do so. Alternatively, the second link I'm giving is to the GitHub repository that includes all the source code for both elude and Percolator. You can also just clone the repository and compile elude from the source code. This is a bit more difficult and requires a bit more computational skills, but there are several instructions available on this webpage. If you encounter any problems with installing elude, please contact the Percolator team and they will be happy to help you out with this. In the next part of this tutorial, I will assume that you have downloaded and installed elude and that you know where the elude executable is located. Now, once you have installed elude, the next step is to prepare your input data. As I explained when describing the workflow of calculating and in silico optimized gradients, you need two input files for calculating such gradients. The first one is a set of training peptides. This is a file that needs to include peptide sequences together with their observed retention time. And these should be sequences that were confidently identified in a run based on a linear gradient. I'm going to show you how such a file can look like. I will just zoom in so you can see better. So this file includes on each line a peptide sequence together with an observed retention time. And these sequences should have been confidently identified in any shotgun proteomics run using a linear gradient. This set of training peptides will be used by the retention time prediction software to train a retention time prediction model. The second file that you need is a file including theoretical peptides. Alternatively, you can also have just a FASTA file including the proteins that you would like, whose peptides you would like to optimize. I can show you how these files look like. This is the file including a set of theoretical peptides. There is nothing special with this file, it just includes one peptide per line, and then there are lots of them. And these are all the theoretical peptides whose predicted retention time distribution I would like to optimize. Alternatively, I can use a FASTA file. And this is the typical FASTA file where you have a protein name and then you have the protein sequence. In this case, I'm going to use, for this tutorial, I'm going to use the yeast FASTA file that will include all the yeast proteome. Once you have these files prepared, the next step is to download the GradientOptimizer software from the link provided here. So I'm just going to copy this link in my browser. And this web page will be displayed. And in order to download the GradientOptimizer tool, I have to click on this green button here that says GradientOptimizer.jar. And I choose to save the file in a convenient place. I will just save it on desktop. As soon as the file GradientOptimizer.jar was downloaded, just double-click this file and the main window of the application will open. As you can see, in the upper part of the window, you need to choose the type of nonlinear gradient that you would like to calculate. In this tutorial, we will look at in silico optimized gradient. In the second panel of the window, you have to give the parameters for the retention time distribution that you would like to optimize. First, we need to give the path to the elude executable. I prepared this file in a convenient place on the desktop. Then we have to give the file including the training peptides. So again, this is the file that includes peptide sequences together with observed retention time that will be used to train a retention time prediction model. I'm going to choose the file that I have just shown you. It was called trainingpeptides.txt. I would recommend using maybe 400-500 of peptides, not more, because that will slow down the retention time prediction method. In the next panel, we need to select the theoretical peptides. As I said, we have two options. Either we give a file that includes one peptide sequence per line, either we give a protein FASTA file and the program will do the in silico digestion, assuming trypsin as the enzyme used to digest the peptides. I will use a protein FASTA file because it is easier. I'll use this yeast FASTA file that includes the full yeast proteome. And if we choose the second option, we also need to give a minimum and a maximum peptide length. So I will choose to consider only peptides between 8 and 50 amino acids long. And this is sufficient to give the retention time distribution to optimize. In the next panel, we need to give information about the linear gradient that was used when the training peptides were obtained. As I said, the training peptides should be peptide identification obtained from a shotgun proteomics run based on linear gradient. And here we have to give information about the linear gradient that was used for generating that run. In this case, the linear gradient went from 10 to 130 and from 2 to 40% B. And also, we need to mention the lag time. This is a parameter that gives the difference between the LC times, the times in which we instruct the LC system, and the raw file times, which is the times that appear in the raw file. If this parameter seems unclear to you, or you would like to know how you can calculate it, once more, please read our paper that introduces nonlinear gradients. We give a clear description of how you can calculate this parameter and what is its meaning. For this particular dataset, the parameter was 6.2. In the lower panel of the window, you can give different parameters for how the nonlinear gradient should be formatted. What is the time step? How often should we sample? 1.0 means every minute. I'm going to use in this run every 5 minutes, and then we can also change the number of decimals that we want in the percentage B, and also we can mention different characters that should be printed before or after the percentage B or the times. Once we set all these parameters, we can just press the calculate gradient button and the software will run elude to train a retention time prediction model, will perform the in silico digestion, will predict the retention time for all the peptides in the in silico digest, and then we'll use this distribution to calculate an optimized gradient that will even it out. What I would like to emphasize here is that depending on how large is your training set and how large is your database or the list of theoretical peptides, this procedure may take a few minutes. As soon as the calculations were completed, a new window will appear on the screen displaying the results. In the left side of this window, you will see the gradient function giving us a list of time points and corresponding percentage B. In the right side of the window, a graphical representation of the nonlinear gradient is given, such that you can get an instant idea of how different this gradient function is compared to a linear gradient. If you would like to change some of the parameters of the nonlinear gradient, for example the time step, how often is this gradient sampled, or the number of decimals for the percentage B, or other formatting option, you can do that in the main window and press the Calculate Gradient button. For example, let's assume we want to sample at every 1 minute instead of 5, and we would like to have 2 decimals in the percentage B. We do these changes here, and we press the Calculate Gradient button. The results will be instantly updated in the new window. For example, now the gradient function will be sampled at every minute, and we would have 2 decimals in the percentage B. Also, the graphical representation is updated. So you can experiment quite easily with different options for the nonlinear gradient and check the results instantly. However, as soon as you change information in the window describing the retention time distribution to optimize, this will require to run elude once more, and this may take a bit longer time. In any case, the results will be always updated in the results window. Once you decide for a set of parameters that are more convenient for you, you can easily save the nonlinear gradient by pressing the Save Gradient button. For example, let's say we save our new gradient on the desktop and we call it in silico gradient. If we will check the desktop, we will see a file called in silico gradient. And I can just zoom in so you take a look. This file will contain exactly the information that we see in this text area in the results window. So this is the nonlinear gradient. Alternatively, you can just copy this to clipboard and just paste it in your ELSE system. And that is it. This is how you calculate an in silico optimized gradient. Just to summarize, first you select the in silico optimized as the type of nonlinear gradient that you would like to calculate. Then you have to give the path to elude a set of training peptides, including sequences, and observe retention time from a shotgun proteomics run based on a linear gradient. Then you can either give a set of theoretical peptides or a FASTA file, including protein of interest, together with the minimum and the maximum peptide length you would like to accept. You then give information about the linear gradient used to generate the training peptides, and you give information about the formatting of the nonlinear gradient and press the Calculate Gradient button. This will give you the nonlinear gradient that will optimize the predicted retention time distribution for the theoretical peptides from the in silico digest. I hope this video was useful for you. I'm looking forward to hear about your experience with using our software. If you're interested in calculating other types of nonlinear gradients, such as the MS1 optimized gradient or a nonlinear gradient based on a custom retention time distribution, please watch our videos describing how you can calculate these gradients. Thank you for watching this video.